So, let's start with the patient, okay? Because I gave the lecture in 2016, and now I'm here, but the patient hasn't changed. And we go out to Tipat Chalav, and we check the kids, and the kid approaches us with an abdominal mass. We palpate an abdominal mass. And this is the renal ultrasound. You see this huge tumor. It's a 3-year-old boy. This is the normal kidney. And the kid has a Wilms tumor. Wilms tumor is a pediatric renal cancer. It could be worse. You tell the parents that most of the chemotherapy and radiotherapy work for Wilms tumors. But I have to tell you, 15% of the cases relapse and they harbor bad prognosis. This is one thing. The second thing is that we put massive chemotherapy and radiotherapy on these patients. So we have a lot of secondary effects of the chemotherapy and radiotherapy. So we have two main challenges in Wilms tumor treatment. One is as I told you, the cancer relapse and the poor prognosis. The other is what we're imposing on these patients. For Wilms tumor, there's another main sequela. This is the sequela. Look, it's a tumor, so we need to take one kidney out. But sometimes if he has a WT1 mutation, a Wilms tumor 1 mutation, the tumor will arise in both kidneys. And this tells you that the surgeon would need to take one kidney out and then about half of the other kidney. So the boy is left with a remnant kidney, and he succumbs to chronic renal failure with time. So this is chronic renal failure. So this is a major challenge. Wilms tumor patients presenting as chronic kidney disease is not such a big challenge. Most of the Western world has chronic kidney disease because he has hypertension, he has diabetes mellitus, and you lose your nephrons due to this huge clinical problem. I'm a pediatric nephrologist. I see kids with renal disease, but the adults see the hypertensive and the diabetic patients. Frustrating. No treatment. You see the patients that have chronic kidney disease go into end-stage renal disease right here. And as you know, end-stage renal disease, we have two main options. One is to turn them over to a transplantation department, or the other is go into a dialysis program, which is quite harsh for a kid. 4 times a week, 4 hours, pretty tough. So we need, for the Wilms tumor patients, to get better understanding of kidney regeneration. Now, this is a new MD PhD student in the lab. He's a graduate of the graphic departments of the IDF. So he hasn't generated a single result yet, but he generated this slide, which shows us the Deco lab with regenerative medicine, trying to regenerate kidneys, and at the same time trying to target cancer, trying to relapse cancers, with mainly focusing on developmental lineages of pediatric tumors and targeting them. And I'll elaborate first on the kidney regeneration part, because it has a lot of relevance to patients not only harboring Wilms tumor. And this is our cell-based strategies for renal repair. As remember, you take out the kidneys, so you have a source of cells. You can discard the human kidney and put it, but when surgeon takes out the tumor, it's a nephrectomy and a partial nephrectomy, you can get a lot of normal kidney tissue. So it would be interesting to start with human adult kidney cells to regenerate the kidney, the remnant kidney of the patient, and specifically renal disease. So what we do in the lab with human adult kidney cells, we reprogram them to an earlier lineages that would confer clonal proliferation and clonal growth to high passages. I'll show you a bit about that. We run clonal assays with the human adult kidney cells, primary clonal assays, or clonal cultures. We grow these adult kidney cells as organoids, as 3D structures, in order to afford a better kidney regeneration. And I'll show you a bit about that. We also use human fetal kidney cells to regenerate diseased kidneys. And I don't have the slide. We use IPS as material, but then you would need to differentiate the IPS to kidney. So more practical in our lifetime would be to use the discarded material, the human adult kidney cells. But when you think about kidney growth, this is not cancer biology. This is just kidney growth. This is, we know that we don't generate nephrons in adulthood. We are born with a fixed number of neurons and a fixed number of nephrons. So we get about 1 million nephrons, and we kill them throughout life. But the fish does so. It really, you can regenerate whole new nephrons in adult life. The mammal doesn't do so. But what we do is this. This is a scheme of a nephron here. And what we have these clonal progeny, which are segment-specific. They arise from different segments of the nephron. This is, for instance, the proximal segment. This is the loop of Henle. This is the distal segment. This is already the collecting system. And this local regeneration scheme by Wnt-responsive clones in specific areas of the kidney compensate for the loss of about 70,000 epithelial cells per hour. You know, we urinate our kidneys out, so we need a cell replacement mechanism. So to say that the adult kidney is post-mitotic is far from truth. This is a proliferating organ that you can really use its characteristics in vivo to grow a bunch of adult kidney cells ex vivo that could afford your patient or anyone with kidney disease with cell replacement. And these are the clones that grow in the adult kidney. This is the blue clone. For instance, it comes from a specific area. These are segment-specific. There's no multipotential stem cell in the adult kidney, just a bunch of committed progenitors. So this is, we wanted to sort of mimic this in vivo adult kidney cell growth ex vivo, and we generated this huge enterprise of clonal growth of primary human kidney from fetal cells and from adult cells. I just want to show you the data where you can take one single adult kidney cells and go up to 5 times 10 to the 6 cells, 5 million cells. So this is the clonal capacity, and you can run up to P5 with just one single cell. So these are colonies derived from single human kidney cells that maintain this called a primitive epithelial phenotype. But we want more cells because we want to replace a lot of diseased kidney cells. You know, when you have CKD, you lose a lot of renal cells. So we want to replenish for these renal cells. So what we do, we overexpress an OCT4. We take an OCT4 gene and we overexpress it in our cells, and we induce partial reprogramming. Now, partial reprogramming is not getting an IPS. It's not the Yamanaka story. It's just getting an earlier phenotype locked in within your same lineage. So this is the OCT4 story. And here, when you generate these long-term self-renewing, you call them IRPCs, induced renal progenitors within OCT4, you can get your clonal cultures going up to P15. This is a primary human kidney. So you get clonal cultures at P15 with a stable karyotype and stable growth. So if we want to get more cells, more clonal cells, more clonal progeny, you would need to induce your adult kidney cells with an OCT4. Okay? So we have the clonal cultures, we have the OCT4-induced progenitors, but this is, I think, this part is the most advanced part in going into the clinic, hopefully. Here you take the kidney, you separate it into single cell suspension, and you do this massive expansion. Now, primary cells deteriorate when you grow them in vitro. They deteriorate, they differentiate. So what we do, we generate organoids or spheres in cultures, and this is how it looks. This is the 2D culture, the expansion phase. This is the turning of the 2D culture, what we call the human kidney epithelial cells 2D cultures, into the 3D cultures, and then you get this self-organizing condensate that grows very insidiously. And you can see standing for HLA, human Ki67, slow proliferation, but you have distal EMA and CD13 proximal tubule markers of the kidney. So this is a method to grow mini kidneys ex vivo just by getting the right three-dimensional context and the three-dimensional culture. Now, you take these cells, these nephrospheres, you put them into mice. And what you see, you can generate a massive tubular network. This is human tubular network in the mouse. This is what we call the in vivo renal potential of the nephrospheres. The mesenchymal stem cells, they don't have renal forming capacity, the fibroblasts, of course. So this is very specific. This in vivo renal potential is very specific to the 3D organoids we culture in the lab from the adult human kidney. And this is just CD13 plus proximal tubular structures. You can see them here. So you can generate very nice proximal tubes. Now, another experiment, we want to take these cells from a CKD patient, from the Wilms tumor after operation. So can you generate nephrospheres from a CKD patient? You can generate them, you can inject them, and you can see this massive replacement of diseased kidney tissue with human nephrospheres derived from end-stage kidneys. So if you take out your kidneys, okay, and they're called end-stage by the urologist, know that they have functional cells that you can cultivate and grow as nephrospheres that one day could replenish your own kidneys. And this is the data. This is human cells. These are EMA, again, distal tubes, CD13 proximal tubes, exchanging or implanted in the mouse, generating this tubular network and replenishing lost kidney tissue. This is the 3D movie because I showed you the sections, but actually you generate these long tubes that regenerate the kidney. And this is when you go to the confocal, you do Z-stacking and you do some bioimaging. You can get these structures that have depth and they self-organize after you inject them into mice. So this is how it looks. Oops. But what about functional aspects? We take we have a model just like the Wilms tumor patient. We take the mouse, we do the 5/6 nephrectomy, then we do the contralateral nephrectomy, and we left with the remnant kidney. And we get the adult kidney cells, and we generate the nephrospheres, and we inject them, we transplant them into the into these nephrectomized mice. These mice with chronic kidney disease. And this is the transplantation protocol where you have transplant 1, transplant 2, transplant 2. This is transplantation of human nephrospheres into a mouse model that mimics progressive renal disease. And this is the functional data. This is the repeated measures model where you can see the trend for salvage of the kidney. It doesn't happen after one shot. This is a pathology of CKD. So you need to put in the protocol says 3 different transplantations into the 5/6 nephrectomy mouse. This is the first injection. This is the second injection, post-third, post-third, post-third. And then you can analyze the creatinine levels and the creatinine clearance in these mice. And you can see functional benefit with treated versus untreated mice. This is human nephrospheres afford renal functional benefit over time. Now, this tells you that you can intervene with the progress of chronic kidney disease. We have the Wilms tumor patient. He develops chronic kidney disease by implanting renal progenitor cells prior to the patient going into end-stage renal disease. And this protocol, the nephrosphere protocol, this here, it's been licensed to a company called Kidney Cure, and the company is heading towards a clinical trial with your own cells, growing your own organoids and putting them back into your kidneys. This is the most advanced, I would say, program in the lab. So we regenerated the kidney, right? At least we regenerated the mouse kidney with the human nephrosphere cells, and hopefully the CKD patient. But what about the relapse? And Wilms tumor, I have to tell you, looks like a developing kidney. This is the normal fetal kidney development, and this is just this is how a Wilms tumor looks. It has this blastema. It has mature figures looking like proximal tubules, and you have some stroma. So it's sort of abnormal nephrogenesis. This is what a Wilms tumor is. Now, and this is fetal nephrogenesis. You have to appreciate fetal nephrogenesis in order to understand Wilms. These are the cap mesenchyme cells. These are the nephron stem cells, which generate your nephrons. They differentiate to pretubular aggregates, and then you get the adult nephron. Now, once you generate the adult nephron, these developmental genes go away. So, if you want to target a pediatric tumor, target the developmental lineage that goes away in the tumor, because it doesn't express in other parts of the body. So it's a perfect, it's a perfect scenario to target, specifically target antigens which lack tissue expression. And I want to mention this. We've been talking about immunotherapy, but remember, pediatric cancers are devoid of mutations. The MRT, I'll show you data, it has hardly mutations, MRT. So I would say the expectation is that the immunotherapy will not be as beneficial as in pediatric cancers as in adult cancers, especially in regard with the mutation load. So we had this, we worked about a decade to sort of show biomarkers of developmental human renal stem progenitors, these cap cells, and Wilms tumor blastema. And these are the transcription factors. But what happened is that these were microarray experiments back then, and then we turned into RNA sequencing. But importantly, this is genes, transcription factors that we specify the renal stem cells in development, and also Wilms tumor blastema. But you need these guys. You need the surface markers if you want to sort out renal stem cells from the fetal kidney, and you need surface markers if you want to really take out the Wilms tumor blastema or understand anything about tumor lineage or targeting specific molecules in a Wilms tumor, because it's really hard to target a transcription factor, right, with an antibody. So these are the surface markers that we could link to renal stem cells and Wilms tumor blastema, such as these were ANCA, this has been published, ANCA, DILC1, activin receptor 2B, and so forth, FRIZL7 of the Wnt pathway. But ANCA is crucial, and luckily, based on these earlier works, we were able to do two experiments. One is to take a human fetal kidney, culture it, and use the ANCA molecule to sort out human fetal kidney stem progenitor cells. Now, we know by now, we started off with an NCAM1 sort, okay? We sorted out the fetal kidney stem cells with NCAM1, but now we need to do negative selection to get them with epithelial markers such as EpCAM1 and CD133, just like hematopoietic stem cells, okay? 3 minutes, oh my God. You sort out a positive marker, and then you sort out a negative one, and you can get your human fetal kidney stem cells out. Skip this. What ANCAM did to us, it afforded the isolation of human fetal kidney stem cells, but also we could get out the tumorigenic fractions or the cancer stem cells from a Wilms tumor, and this is past work where we took out 200 ANCAM plus ALDH1 plus cells and sorted them out from a tumor, Wilms tumor. These were Wilms tumor xenografts, because it's not as easy to work with pediatric cancers as with colon carcinoma for sorting experiments. These are the 200 ANCAM plus elite cells that generate tumors in mice, and you can really, this is a tumor fraction which has self-renewal. You can go to very high pathogens just by sorting out this fraction, regrowing it into mice, resorting, and you can passage the tumor just by sorting out these cells. And I just want to show you, this was the data with the anti-NCAM1 antibody, where we knocked off Wilms tumor. This was 60 mice, and we collaborated over the years with the Sachi Fainaro lab at the medical school of Tel-Aviv to generate a polymeric carrier with paclitaxel with an NCAM targeting peptide, and we could show that targeting NCAM would be beneficial in a Wilms tumor. And actually, our work ignited this phase 2 study of lorvotuzumab martensin. It's an anti-NCAM1 compound in children with relapsed or refractory Wilms tumor. So the results will come out in 2019. I hope it will be helpful to these Wilms tumor. It's relapsed, okay? Because chemotherapy and radiotherapy are good for Wilms tumor. So this is relapsed Wilms tumor, and this is the phase 2 that luckily was ignited by the basic findings in our lab. So this is a very nice example of bench to bedside. But life is complicated, and I want to show you Rna Seek data of the tumorigenic fraction we sort out of Wilms tumor and the non-tumorigenic fraction. And this tells us that, life is, if we target with the anti-NCAM1 antibody, we might not get, the results won't be as good as we expected. This is the Rna Seek data. This is the positive fraction. This is the tumorigenic fraction. This is the less or the non-tumorigenic fraction. And you can see this is a xenograph. So these are presumably mouse cells that have low alignment rate to the human genome. I just want to show you that these cells look like uninduced mesenchyme, but they are both the tumorigenic fraction and the non-tumorigenic fraction lay in this cluster. But the tumorigenic fraction is actually here. This is the tumorigenic fraction. And when you look at these cell types, they express genes related to muscle development, myosinins, actins. SNE1 is high. This is epithelial to mesenchymal transition, meaning that the tumorigenic front of Wilms tumor lies in a lineage that is even differentiated from the earliest renal lineage, the uninduced mesenchyme. So this is the mesoderm, and it probably comes from a lineage which is early muscle, which is earlier than the uninduced mesenchyme. This means that NCAM1 would probably downregulate once cancer progresses. Okay, and the anti-NCAM1 story, just by reverting to an earlier embryonic lineage, might not work, but let's see. This is just two populations that interact through these. This is the DLL1. This is the Notch2 pathway. We're trying to look at the relationship between the tumorigenic and non-tumorigenic fraction. We expand the scope in the PDX family farm we have at Shiba. Well, we've been able to discover, by just growing tumors, sorting out fractions, molecules that are important for tumor progression, such as PPR gamma for renal angiomyolipoma and leusiloxidase for malignant rhabdoid tumor. And this is just, I don't have data, just go through this, don't have time. This is the recent paper by Oren in the lab, the PPR gamma antagonism. But, I'm good with time? But the message is this. For the Wilms tumor patient, put kidney cells aside, regenerate, grow them, generate organoids, put them back, hopefully, to regenerate his remnant kidney. For the relapse, we afford anti-NCAM1 immunotherapy. Let's see the results in 2019. The data, the Rna Seek data predicts that there is a tumorigenic, I would say, front that is differentiated to an early lineage and that might escape the anti-NCAM1 therapy. So let's see. And this is the people behind the work: Orit Steinberg-Harari, Nomi Podeshaked, Eina Vax. Very old slide, and I need to refresh it. And I want to thank you.